Welcome, everybody. It's that time of year again where we get to hear the great work that our IDSC Fellows are doing. This year we have 2 atmospheric scientists and 4 from the life sciences doing work on using machine learning and data science to advance their fields. So we will start with the youngest member, and I grouped this by
Birthday?
topics.
No, oh.
And I left the longest presentation to the end, but please leave it to about 15 minutes. And mind you that we, leave a little bit of room for questions and answers. And we will end with a little bit of networking events with light refreshments. And our first speaker is Nimay Mahajan, and I will give you the floor. Thank you.
Awesome. Hi, everyone. My name is Nimay Mahajan. I am a current junior here at the University of Miami, double majoring in both meteorology and mathematics. Alongside my mentors and advisors, Dr. Ben Kurtman and Dr. Mary Beth Arcodia, I've been exploring the dynamics of a changing climate and more specifically analyzing the Indian monsoon region, as well as the Atlantic Ocean and their interactions under elevated carbon dioxide conditions. Just to get into a quick little motivation as to why I'm doing this research. In general, climate teleconnections between 2 specific regions play a crucial role in understanding and predicting global weather patterns. And so in specific, this study investigates the relationship between atmospheric dynamics regarding Atlantic hurricanes in the main development region, which is this region right off the west coast of Africa, extending into the Caribbean islands, as well as their connection to monsoon variability in the Indian monsoon region. Specifically, we hypothesized that entropy or vorticity squared anomalies in that main development region may be a predictor for precipitation anomalies in the Indian monsoon region. And to kind of touch on that a little bit more, vorticity is a measure of rotation. And so the reason we had to square this value was because negative vorticity and positive vorticity both indicate rotation. And so that sign dependency was something we had to get rid of. And so squaring that value provided us with the entropy that we decided to analyze. And it's very clear that understanding this teleconnection can help improve seasonal forecasting and enhance our knowledge of large-scale atmospheric circulation patterns in general. So moving on to my methods, to be quite frank, the data pre-processing for this research took up the majority of my time. This is the first time I've worked with such a large data set, and so kind of understanding how to smooth and get rid of the noise within my data was vital for me. So first, I extracted the entropy anomaly data that I had, as well as the precipitation anomaly data from the IMR. I then applied both temporal and spatial smoothing. So first, temporally, I used a 10-day running mean. And this helped kind of filter out that high frequency variability that would add noise to our data set. And then I also applied spatial smoothing through Gaussian filtering. And this helped once again reduce that noise and highlight those large scale patterns that we were looking for. And then lastly, for the pre-processing, I had to standardize the variables. And so entropy has units of 1/seconds squared, and precipitation was measured in millimeters. And so getting rid of that unit dependency was important. So dividing by their respective standard deviations and normalizing the data was very important. Moving on to the analysis, we conducted lagged correlation analysis. And so this basically took the mean of both regions and correlated them together to understand the lag between the 2 regions. And so when something happens in the MDR, we can't expect that would immediately happen in the Indian monsoon region. And so we found a lag of about 5 days. Okay, all good. I then calculated the cross covariance matrix. And so this was vital as compared to a normal covariance matrix, because you're allowed to look at variables in 2 separate regions at once and then correlate them together and understand the varying nature of them. And then I applied singular value decomposition or SVD to help extract these coupled modes of variability. And so the SVD provided me with 3 separate matrices. The left singular projection was my MDR projection that I used to plot my EOFs on the next slide. And my right singular projection was for the IMR, and then the middle matrix was there to understand the power of each mode. It is also important to note that I did focus solely on hurricane season, and so this was from June through November to isolate, just specifically this season and understand how hurricanes in specific could be a predictor. And so here are the EOFs that I ended up creating. And these were derived from the cross covariance between the MDR and the IMR, like I mentioned. In general, these patterns represent the dominant ways that variability in the MDR and IMR are connected. And so at the bottom of the screen, you'll see the percent variance explained by each mode. And you can see that in both experiments, both the normal and the doubled carbon dioxide experiment, the first and second modes were the most important. And so they both, in the first experiment, explained about 73% of the variance, and in the second one, explained about 66%. And so we decided to really just narrow in on those 2 modes and analyze them. It is also important to note that with SVD and with these projections, we do have this arbitrary sign ambiguity that comes with EOF eigenvectors. And so to get rid of this sign dependency, I regressed the time series onto the original data. And so this gave us these regressed anomaly maps that you see, where they're physically interpretable now, and they're consistently oriented across both experiments. And so we don't have to worry about that sign flip anymore. And so specifically looking at just the normal experiment, we can see that the MDR pattern, which is on the left side of your screen in mode 1, is particularly diffuse, and it's relatively disorganized, and this coupling is relatively weak with the Indian Peninsula. We don't see a strong indication of higher precipitation anomalies in this mode. But mode 2 is a little bit more interesting, which we'll see on the next slide as well. Mode 2 of the normal experiment actually ended up being very similar to mode 1 of the doubled carbon dioxide experiment. And I thought this just implied that this sort of teleconnection mechanism could become more dominant in a warmer climate. And so something that's already present may be worth more in a stronger environment, and may be weaker and have less of a signal right now. And so this can obviously have prediction insights and give us insights into how these patterns might be shifting under a changing climate. And so moving on to my doubled carbon dioxide plots, we can see that this coupling does seem to be a little bit stronger across this experiment. In the MDR in mode 1, we can see this kind of northward shift of entropy anomalies. I personally associated this to a possible shift in the ITCZ, and that has been supported through climate research that the ITCZ may be shifting, and this directly impacts the African easterly jet, and subsequently the tropical waves that come off the west coast of Africa. And so seeing this northward shift could have implications to the weather that we see across the Caribbean and throughout the US as well. But we can see that that's linked to negative anomalies, particularly over the Indian Peninsula. This is kind of a disruptive signal as compared to what we saw under the normal experiment. And so this is definitely something that could use more research. It also could explain the expansion of the Hadley cell or changes in Rossby wave propagation in general. Moving on to mode 2, we see this dipole structure in the MDR where we see these strong positive anomalies over to the western side of the MDR now, with this negative anomaly off to the eastern side near the coast of, or the western coast of Africa. And this kind of relates to a increased precipitation trend over the Bay of Bengal, which I thought was really interesting. This is a region that typically under monsoon conditions don't see the greatest impacts as opposed to the West Coast. And so seeing this shift in rainfall coupled to the MDR could have big predictive insights into how that region of the world and specifically the eastern coast of India may have to prepare under a changing climate. This once again could also be due to shifts in the ITCZ, and so I think as part of future research, this would definitely be something that I look at in terms of how the ITCZ is shifting and how that's shifting our rainfall patterns there. On top of the plots, I wanted to kind of plot the time series as well to understand how these variables correlated. As you can see by the faint lines in the background, that was the original data with just the Gaussian smoothing added to it. But the data still remained relatively noisy. And so to kind of understand that subseasonal relationship that I wanted to portray, I added another 30-day running mean on top of this to smooth it even more. And we can see some specific areas of these time series that are pretty well correlated, further demonstrating this teleconnection mechanism, and it does help us understand that our lagged correlation that we did conduct earlier in the research was relatively successful. And so all of this research kind of led me up until this point. I was able to present at the Rosenstiel Undergraduate Research Symposium last Friday, which was a great experience. This was my first time ever making a poster and presenting it. So definitely a great and unique experience for me. A huge thank you to the IDSC Fellows program for providing me with this platform. In terms of future work, there was an attempt to implement machine learning to this project, but I did come across severe data leakage, and this did lead to misleading results. And so as time goes on and I have more of an opportunity to continue this work, this is something that I definitely plan on looking at. As I presented my poster last Friday, I also looked at or took advice of others who came up to my poster, and a big consensus was isolating El Nino and La Nina years to see if these signals vary. So that's definitely something that I want to look at. Another thing is ocean-atmosphere interactions play a major role. And right now I'm only looking at atmospheric relations with entropy and precipitation. And so maybe adding sea surface temperature and other ocean-based impacts could benefit this research. As I mentioned before in the previous time series slide, there were specific times where the relationship really did seem to correlate very, very well. And so I thought it would be beneficial to kind of concatenate those time periods together and really dial in on those times in which the correlation was very strong to understand this mechanism better. Also, through the poster demonstration, I learned that rainfall fields are extremely intermittent and extremely noisy, and we see this within the data as well. And so other variables that I could use that kind of replace this precipitation and maybe help lead to better results is precipitable water or column integrated moisture flux, and to better grasp that moisture that's present over the Indian monsoon region. Another major variable in climate science is OLR and outgoing long wave radiation and understanding radiative fluxes and so on. And so that could be another variable to help underscore the convective features in the Indian monsoon region. And then finally, wind plays a huge role in the Indian monsoon. And so this research really only focuses on precipitation. And so it would be definitely interesting to add these lower level wind anomalies and see the reversal of winds and see how that's an indicator within this connection as well. Lastly, the last thing that I wanted to point at is that the smoothing was a major issue in this research. And so kind of understanding and learning how to better smooth my data and help highlight those correlations better would be definitely something that I want to look at in the future. Just wanted to give Dr. Mary Beth Arcodia and Dr. Ben Kirkman a huge shout out. They were the greatest of help for an undergraduate like me, and obviously a huge thank you to the IDSC Fellows program for providing me with this avenue. Thanks.
Quick question for Nimay?
Go ahead. Okay. Go ahead, Ravi.
I have a question about the cross covariance matrices. Have you used any linearization methods or what kind of computational complexities or runtime we can talk about?
Yeah, so this was my first time ever computing this. This was a major complication in our research. It actually got to a point where Dr. Kirkman had to send me an equation that I had to end up hard coding into my code. Yeah, outside of that, I've never really used it. So yeah. Thanks.
Don't worry, he sends me equations.
Super well, that's what he does. So, one very quick question. You actually mentioned something about using the machine learning and you have data leakage. And so can you explain a little bit to everyone here? Because I'm not so sure that everyone can understand what you mean by that.
So at first in my model, I was using randomized data. And so what the model was training on data that was already happened in the future. So that was leading to misleading results. So once I changed it into a 70, I think it was a 70, 15, 15 split. The results were pretty inconclusive. And so I tried adding more predictors such as ENSO, MGIO and stuff like that. But any monsoon region has relatively low internal variability. So those factors didn't really play too much of a role.
So when you say leakage means.
Training on stuff that it's not supposed to train.
Correct.
Right? Okay. Our second speaker is also an atmospheric scientist, and she's been trying to apply machine learning to ENSO prediction.
Yes, all right. All right, hi, I'm Cait Martinez. I'm a third-year PhD student in the Department of Atmospheric Sciences. My IDSC mentor for this project was Sam Goldberg, or is Sam Goldberg, and my PhD advisor is Ben Kirkman. When I wrote the proposal for this fellowship, this was the title of my proposal, ENSO preconditioning versus noise as the forecast evolves. And as it turns out, this is sort of more of like a future direction for this project. So I want to talk to you today a little bit about some work I've done developing a machine learning based interactive ensemble for ENSO. And so just a little primer, I guess, on ENSO. That's the El Niño Southern Oscillation, and it plays an essential role in modulating the global climate through its atmospheric teleconnections, like Nimay was mentioning before. And to sort of highlight that, kind of similar to his work that he was doing, this figure from NOAA climate.gov shows the typical influence of El Niño on the top and La Niña on hurricane frequency. And so kind of diving into what ENSO is, it's sort of driven by these interactions that involve the exchange of energy and momentum between the atmosphere and the ocean. And the resulting coupled feedbacks are really what's central to creating and amplifying those events, which, for instance, El Niño, La Niña, and they are defined by this Niño 3-4 index. And I'll get into that a little more later. But so even though ENSO research is been happening for decades, really, there's still quite a bit of debate on which mechanism dominates in ENSO predictability. And so generally, subsurface preconditioning, also sometimes called the subsurface precursor, it's this buildup of anomalous ocean heat content. And that's generally been considered the main source of ENSO predictability. But I think that the role of atmospheric noise remains unclear. And so that's sort of the kind of whole overarching, my PhD dissertation in a line right there. So I'm really trying to look at what role does atmospheric noise play? And so, and how do we, to sort of isolate that noise, a really cool approach is called the interactive ensemble. And this is a coupling method. I think it was first introduced by Kirkman and Shukla in 2002. And essentially it's designed to reduce noise at the air-sea interface. So looking at this, oh, I don't have a cursor, sorry. zoom. Looking at the figure on the left, we have multiple atmospheric component models coupled to a single ocean model. And this is in the context of state-of-the-art, fully coupled circulation models. Now, what happens is these atmospheric models are the ensemble mean is taken of these atmospheric components, and then that ensemble mean surface fluxes are fed to the ocean. The ocean responds with SST, and the SST is fed back to each of those individual atmospheric models. And so this is really cool because it allows us to diagnose mechanisms, which mechanisms dominate and where, and then we can also look at how the noise affects variability and predictability. But a major downside of the interactive ensemble and really just fully coupled models in general is that they're extremely computationally expensive. And so one of the sort of driving points to this project is to how can we make this interactive ensemble more accessible on, say, your laptop or in a Jupyter notebook. And so this kind of begs the question, can we simulate that outcome of the interactive ensemble with machine learning? So what we did was we developed a hybrid convolutional neural network paired with a long short-term memory architecture. This is just a very simple schematic that I put together in PowerPoint of what the model is actually doing. But we have really 4 input features: zonal wind stress and meridional wind stress, the vertically integrated ocean heat content over the upper 400 meters, an ocean mass to sort of help the model learn continental boundaries. And then the target variable is to predict this Niño 3-4 index, which is just the monthly anomalous sea surface temperature anomalies over a small region in the equatorial Pacific. And so basically I decided to use CESM2 pre-industrial control model data. It's 2,000 years, so 2,000 times 12, it's a lot. And because I have this really long time series of all these different variables, offers a really interesting opportunity to train on the first 200 years and then validate or test on 9 separate 100-year windows. So for instance, I train on years 1 through 200 and then test on, say, years 501 through 600, and then maybe 200 years later, what's that? 800, year 801 to 900. And so this is an attempt at preventing data leakage, for instance, which I think I might have had that same problem too. But but it turns out this model is actually okay, right? Like, so this is a figure I made of this first model. You know, I want you to pay attention to just the 2 left panels. So on the left, the very far left side, I've got the predicted SSTA from the model against the actual SST, the SSTA, the truth from the control. And it actually does a pretty good job. I mean, that's an R score of 0.84. That's not so bad. It does look like it under predicts the extreme values on both ends there, on both ends of the tail of the plume of the scatter. And then on the middle panel, we have that same information, but as a time series. So the black is the control actual truth, and the green is the predicted SSTA. And once again, you can see the model doesn't really capture those real extreme values, but at least the sort of ups and downs are they're captured quite well. So taking the model, taking the interactive ensemble, how can we incorporate those 2 together? And so essentially we want to answer that question of how can we simulate that outcome of the IE, which is minimize atmospheric noise at the air-sea interface with machine learning. And we could take an ensemble of the control. You could create an ensemble of it because we have such a long time series of it. But we could maybe say, let's say you have 6 ensemble members, well, you could change the random seed in each of those, but that only measures the model's variability. So we want to make sure that we're only reducing the noise and the wind stress, those atmospheric inputs, and not the ocean heat content. So we kind of put together maybe some noise reduction methods. These are maybe like the low-hanging fruit. I'm sure there's many other more sophisticated methods that you could do for this, but I just wanted to highlight a few of the first three that I've been working on. High-frequency filtering. This is a temporal low-pass filtering, similar to what Nimay did, but because I have monthly data, it's not as low frequent, or it's not as high frequency as I'd like. There's a spatial filtering, and this is just filtering out the incoherent wind stress that's not captured by the first EOF, and what I call a local unstructured filtering. And this is just a simple spatial coarsening to reduce the grid scale noise. So two of these are spatial. One is sort of a temporal. And then and then these are, so I've used, I used ChatGPT to ask what, if it like, describing these three noise reduction methods. I asked it to create some images for me, and this is what it came up with. And I'm not sure I totally agree, but we'll use them there. I thought it was kind of fun. So what does this actually look like? My conceptualization of this in the interactive ensemble. Oh, there we go. Okay, so it's the easiest to understand, I think, if we focus on the ocean components first. So let's say we have N ocean components or N ensemble members. If you have the same ocean inputs and you average it, it's going to be the same ensemble mean. So this is just that this is just not reducing any noise in the ocean inputs. Now for the atmosphere, and we're still using this random seed. Now we're using the random seed in the pre-processing. So that's each time the high frequency filtering, for instance, is being applied, a random seed is being applied on each of those six or N members. And then when we average them together, that also reduces any residual noise that maybe isn't fully filtered out by those noise reduction methods. And just sort of some preliminary results. I actually have like 30 models running right now on a remote cluster, and I'm hoping to have some updates of this for my my poster tomorrow at Computing Day. But this is just a heat map that shows the ENSO prediction performance measured by RMSE for the various noise reduction methods on the y-axis and various loss functions on the x-axis. And it's clear right here that, more blue is going to be better, less lower RMSE, more red is worse. So clearly the high frequency filtering is the leading contender right now across all loss metrics, but it looks like right now the Huber loss, which kind of is it's an L1 loss, but it is kind of almost like an in-between between an L2 and an L1. And that seems to work pretty well for ENSO, capturing ENSO variability when predicting that Nino 3-4 index. And so that's kind of what's doing the best right now in terms of my models. But I still, like I said, I'm hoping that this plot will be able to be updated this evening and we'll have a little bit more of a better feel. There may be only a few ensemble members in in the spatial filtering or in that local unstructured noise. I think the high frequency filtering worked the best so far, or has the most ensemble members finished. So sort of cycling back then to that first slide, these are sort of the future directions that I want to take with this project. I'm not done with this project. It's actually becoming a huge part of my PhD work. But like I said before, one of the cool things about that interactive ensemble is that we can diagnose coupled feedback strengths with SST variance ratios. And so we can sort of look at, all right, where are unstable coupled feedbacks important? Where are nonlinearities important versus the atmospheric forcing? And one of the cool things about machine learning that we can use is explainable AI. So like a GradCam, for instance, method to identify the relative importance of deterministic ocean versus the stochastic atmosphere in reducing the prediction errors. So the model says, hey, I think that this is, I think that maybe these areas where they're blue in the wind stress, that seems to be more important than these areas in the red in reducing those prediction errors. And finally, that like going back to that first title slide, my whole goal is to diagnose the relative role of atmospheric noise as the forecast evolves. So I'm brainstorming ways to use this hybrid model in some of my predictability experiments or my predictability ensembles that I have. I haven't quite figured out exactly how to incorporate that yet, but it's something I'm excited about. And with that, I'd just like to thank NSF, NCAR, obviously IDSC, the University of Miami Rosenstiel School, my PhD advisor, Ben Kurtman, Sam Goldberg, Mo for kind of like rallying us and keeping us on track all semester, my husband, Bubby, and my family, and some my friends, Rachel, James, Michael, and the rest of the crew at Rasmus. And I will invite you to connect with me over email or on my website. Thank you.
Questions for Cait? Ravi?
In your ensemble models, have you said like you use GPUs, right, for all the machine learning?
Oh, I'm using CPUs. I'm not using GPUs.
You know,
I want to. Ben advised me that I might spend more time trying to get it to work on GPUs than it's probably worth. But it is something that's in the back of my mind. I'd like to.
because there are two things that you can look at.
Mhm.
One is the speed up of the ensemble. Of course, maybe Ben told you because these ensembles require the, what do you call it? Boundary matching requirements or statistical covariance to look at differences between different ensembles and their connectivity. That's all non-linear problem. GPU may not solve it. But what I was really looking at is we have these new GPUs that you can, they're very powerful. So you can at least try them to see how they compare with your traditional CPU calculators.
Is this on Triton?
Yeah, it's a Titan, but you can. talk
It's a
to
different
about,
cube.
Oh, because Triton scares me.
There is a new testbed cluster that we have
Okay.
with the most sophisticated GPUs and CPUs. I think, I don't know if this is the code, but Ben ran the test on the testbed and he got about 54x speed up.
Oh, wow.
So I'm really interested because the new latest GPUs actually support multi-job configuration, so you can really distribute the ensembles across the GPUs and get things done. It's very similar to CPU, but probably it will add more.
That would be great. I mean, that's how I run. I'm familiar with it because that's how I run my CES. When I run CSM2, I run it on Derecho. And so it would be great to like just speed up.
The second question is follow-up is, when you ran the ensembles, how did you try to lean to, how do we put it? When you run the ensembles to simulate the characteristics of the ocean conditions, let us say, do you normalize them at each step to see how much information they gather and then before you go to the next one, or you just independently iterate each model?
Oh, so are you saying, I feel like my little schematic might have been misleading. So the data that, are you saying in the pre-processing of the,
In one of
Sure.
the workflows, you had several ensembles that are connecting into.
no. Oh, there we go.
Keep going back, yeah, that one. Next one.
Mhm.
Next, yeah, this one.
Yeah, this figure's a little misleading, right? Because, I have that like double arrow there. That's not really, it really probably shouldn't be there, right? So really what it is all of this moves forward in time. I have all of the wind stress through 2,000 years, and as well, where I calculated the ocean heat content through the 2,000 years. And so the filtering happens on a different random seed per member for each of the however many members. And so it's not really actually talking to each other. It's really just
So how do you know when to stop the calculation if you're not really looking at the tolerance between the models?
well, I mean,
They're synchronously integrated in time. They're just being averaged when they have to couple the
Yes.
ocean and the atmosphere.
Okay, so there is some talking between the ensembles, okay. All right. thank you.
kind of, yes. So, and I'm hoping, I still have to make my post here, but I'm hoping that I'll have some really cool results to show tomorrow. So.
Okay, tomorrow more questions.
Tomorrow. I'll send them tomorrow. Thank you.
All right, thank you.
Thanks, Cait. Okay. Our next, we're changing gears a little bit, going towards the life sciences. Jessica Okutsu is our next speaker. She's trying to use machine learning to study if cadmium can be used to cure zebrafish.
Thank you. So, hi, I'm Jessica Okutsu. I'm from the biology department. My PhD dissertation is on potential neuroprotective effects of selenium against cadmium-induced photomotor mechanosensory and morphological impairments in larval zebrafish. And this is a data-driven approach to neuroprotection research. Okay, so what if there was a natural occurring essential element that could protect our brains from toxic metals in our environment? Today, I'll share how selenium might do just that and how data science is helping us uncover this protective relationship. My research examines how selenium might prevent cadmium toxicity, a critical environmental health issue affecting communities worldwide. Using zebrafish as our model and behavioral analysis techniques, we're working towards solutions that could impact millions exposed to industrial pollution, contaminated water, and cigarette smoke. Sorry, didn't mean to go forward. So cadmium exposure is a global health concern, particularly for vulnerable populations near industrial sites and in developing regions with limited water treatment. This toxic metal causes oxidative stress, neurological damage, and developmental disorders, effects we see also mirrored in our zebrafish model. Selenium, on the other hand, is an essential micronutrient with powerful antioxidant properties. Selenium, with a recommended daily allowance of 55 micrograms per day, supports immune health and may function as a potential therapeutic against cadmium toxicity by reducing cadmium cellular transport. It's also a vital component of antioxidant enzymes like glutathione peroxidase, which are responsible for scavenging reactive oxygen species, or ROS, and reducing oxidative stress. So our main question investigates if selenium supplementation may serve as a therapeutic intervention against cadmium-induced toxicity if selenium is applied as a prophylactic. This research has significant real-world applications, from informing dietary guidelines and supplementation strategies to guiding environmental remediation efforts in affected communities. The higher cadmium concentrations we're testing match the maximum allowable level in drinking water, making our findings directly relevant to human exposure scenarios. So our 4 sub-questions, which I will talk to you about today, focus on whether selenium mitigates cadmium's adverse effects on morphological development, hatching success, mechanosensation, and vision. This is going to give us a comprehensive view of potential protective mechanisms. And another important follow-up question is, if selenium is effective, which concentration range is the most effective? This is an important question for any therapeutic substance because anything can be toxic at high enough concentrations. In other words, the dose makes the poison. Okay. So in our initial morphology findings, they've been promising so far. Shown here are morphological abnormalities we see in our zebrafish: normal, also known as lack of abnormalities, trunk curvature, tail malformation, edemas, and death. We also expect to see hatching by day 3 days post-fertilization, or 3 DPF, and any later would be considered a delay in development. So we've already demonstrated that selenium pre-exposure provides some protective effects, particularly for hatching rate, tail malformation, and trunk curvature. All are critical developmental endpoints. And we see a complete rescue with hatch rate, as well as a partial rescue for frequency of tail malformation and trunk curvature, around 10 to 50 micrograms per liter of selenium pretreatment. And so although this is helpful information, it would be nice to have more detailed information about overall trends in our data for all concentrations instead of pooled data. Okay. So these visualizations were created with the help of my IDSC mentor, Elvis Maradzike, and they represent a significant analytical step forward. They allow us to evaluate effects by specific concentration levels rather than broad treatment categories. And the distribution plots show binary outcomes, presence or absence of morphological abnormalities across different selenium and cadmium concentrations. So beginning at the upper left grid and working our way down to the right, we can see how hatching success, normal development, tail malformation, and trunk curvature vary across treatments, which are listed on the x-axis, and the y-axis shows frequency of occurrence. The bimodal distributions with peaks at 0 and 1 indicate the binary nature of these outcomes. Either a fish exhibits the trait or it doesn't. 0 means absent, 1 means present. What's particularly striking is the pattern across treatments. It seems that moderate selenium levels, particularly 10 to 100 micrograms per liter, appear to provide the most protection against cadmium-induced abnormalities. This is especially evident in the normal development N plot, where certain selenium pretreatments shift the distribution toward 1, indicating normal development, even with the subsequent cadmium exposure. We can also see that certain selenium levels, also around 10 to 100 micrograms per liter, appear to shift the distribution towards 0, indicating no tail and trunk malformations. So we can also see that some of the selenium-only controls did so well that they outperformed our lab controls. So in this second grid of morphological data, we're examining edema formation mortality. E stands for edema, PE stands for pericardial edema specifically, an umbrella under edema. And those are common developmental abnormalities affecting cardiovascular function and overall development and quality of life, as shown in the image on the left, where you can see the swollen yolk and pericardial edemas in a cadmium-exposed larval fish. The D row represents mortality, shown in the bottom image of a 5 microgram per liter exposed, a cadmium-exposed larval fish that did not survive to 5 days old. The bimodal distribution again indicates the categorical nature of these outcomes. So looking at the edema plots, we see that the selenium pretreatment, the pretreatments modify the occurrence of all types of edema, including pericardial, but with varying effectiveness across concentration ranges. The pattern observed in the previous slide continues here, where we see a moderate selenium concentration of 10 to 100 micrograms per liter is showing us the greatest protective effect, while the higher 150 to 200 micrograms per liter of selenium shows much less protection. It appears that we have an optimum dose range of 10 to 100 micrograms per liter for selenium that trend throughout all of our morphological endpoints. So now we will shift our focus to the behavioral data. So the images you see show representative zebrafish embryos, larval fish from our control, cadmium only and selenium, followed by cadmium treatment groups. So through high throughput behavioral tracking, we're able to capture movements at the 0.02 second frequency. Each of these fish is 1 in a 96 wall plate, and timing is sped up by 48 times. Do you see differences among the control cadmium and selenium to cadmium treatments? So I have observed the cadmium alone fish tend to have like quick bursts of movement compared to the control and selenium to cadmium treatments, but that's my opinion. That's why we're doing data science. Each fish's response to light or dark transitions and mechanical stimuli is recorded and associated with those morphological traits that I talked about earlier. We do that for every fish. Perhaps certain concentrations of cadmium or selenium provide certain characteristic movements in these fish. So here we visualize raw tracking data from our mechanosensory tests. And each colored line in the top left graph shows us movement over time in our raw data for test treatment combinations. At approximately 30 seconds, at the time point 6:30 shown with the hand icon, we deliver a tap stimulus and track the resulting movement. From that data, we created the graph to the right that shows a violin plot of distance moved after tap stimulus by pool treatment groups. Violin plots combined aspects of, box plots with density distributions showing both the distribution of data points and their probability density. The letters A, B, C represent statistically distinct groups based on an ANOVA statistical test, followed by Tukey post hoc analysis. And when two groups share a letter, that means they are not significantly different from each other. We found that the control and the selenium to cadmium treatments both have letter A. They show similar responses, and whereas cadmium only and selenium only showed B, which was interesting. This may suggest that selenium pre-treatment rescues mechanosensory function, but this is with all treatments pulled together, so it's hard to say. In contrast to the mechanosensory data, our white light stimulus data shows no recovery to light stimulus in fish pre-exposed to selenium. This is also a critical finding because it implies that there are certain neurotoxic mechanisms that are not rescued by selenium that are rescued maybe by mechanosensation, in mechanosensation. So to make sense of our behavioral data, we conducted principal component analysis across 9 tracking variables from both tap and light stimulus tests. The first 3 principal components captured approximately 92% of the total variance. These tracking variables were a mix of either total distance or velocity before and after tap stimulus or light on and off. Looking at the first 2 principal components, which together explain approximately 80% of total variance, we can see how different behavioral measures relate to each other. PC1 is 57% of variance, and it primarily captures overall activity levels with variables like distance traveled and velocity having strong positive loadings. PC2 is 20%, accounts for 20% of variance, and it differentiates between baseline activity and stimulus response. So yeah, the color indicates squared cosine, sorry, which measures how well each variable is represented by a component. Brighter green indicates better representation, and black represents worse. So what's particularly interesting is that the variables associated with white light response highlighted in these boxes have consistently lower contribution to the principal components, compared to the mechanosensory variables. And this aligns with our earlier finding that selenium rescues mechanosensory function, but not vision-related responses. The clustering of mechanosensory variables suggests that these measurements capture related aspects of the same underlying neural processes, which appear to be differently affected by cadmium toxicity and selenium protection. The nearly perfect overlap between mean velocity and total distance measurements indicate redundancy, suggesting we could probably streamline future analyses by just eliminating one of them. Oops. Oops. Okay. Technical difficulty. Okay. So to identify natural groupings in our behavioral data, we applied k-means clustering to the principal components extracted earlier on the behavior tests only. This was unsupervised learning, and it allows us to discover patterns without treatment information. But first, we needed to figure out, we selected 4 K clusters. I put this on the wrong slide here, based on Hartigan's rule of diminishing returns of adding more clusters. Oops. Oh. Did this not? Sorry, a little out of order here. Don't know where the other slide went, but we'll look at the actual data here instead. So, unfortunate. So let's see. So here we overlay the actual treatment information on our predicted clusters. The different symbols represent different treatment types: dots for cadmium, triangles for control, squares for selenium, and plus signs for selenium to cadmium transfers, while colors still indicate the predicted clusters. So the table at the bottom right shows the distribution of actual treatments across predicted clusters. Groups 2 and 3 predominantly contain control and selenium only points, which represent healthy phenotypes. Group 1 and 4 capture more of the cadmium and selenium to cadmium transfer treatments. This partial alignment between unsupervised clustering and actual treatments partially validates our approach. The fact that some selenium to cadmium treatments cluster with controls while other cluster with cadmium only treatments suggest varying levels of protection depending on concentration and exposure protocol. The overlap between cadmium and transfer treatments in cluster 1 and 4 suggests that while selenium provides some protection, the behavior of cadmium exposure persists to some degree, perhaps. So this does demonstrate that behavioral phenotyping can distinguish between treatment conditions with reasonable accuracy, suggesting potential applications for automated screening of both toxicity and protective effects in future studies, with additional supervision and testing, of course. So our findings demonstrate a hormetic effect of selenium on cadmium toxicity, meaning a dose response relationship where low and moderate concentrations, 10 to 100 micrograms per liter, provide optimal protection, while higher levels, 150 to 200, can themselves be toxic or even exacerbate cadmium's effects potentially. And this is crucial to better understand the therapeutic window for selenium supplementation. The protective effects were most evident in the morphological endpoints and mechanosensory function, while vision appeared unresponsive. This selective protection suggests different neural systems and shows that there may be varying susceptibility to both cadmium toxicity and selenium's protective mechanisms. Our PCA revealed that behavioral responses to mechanosensory stimuli contributed most significantly to the data variance, with light response variables showing minimal contribution. This confirms our need to elucidate the differences between visual and mechanosensory neural mechanisms in zebrafish moving forward. The K clustering showed promising ability to identify treatment-related behavioral signatures, though refinement is needed to improve classification accuracy and identify optimal selenium treatments moving forward. If we could definitely focus on like one treatment, that would be ideal. This work is an exciting intersection of environmental toxicology, which is what I have a background in, neuroscience and data science, and it has the potential to impact public health across diverse communities. This IDSC fellowship began at the start of the semester, and time has gone by really fast. If we did have more time, I would love to delve deeper into my data set. And that brings me to some next steps. While there are some valuable insights into selenium's predictive mechanisms, there are several advanced data science approaches that could further enhance our understanding and predictive capabilities. First, we'd like to incorporate morphological data into the clustering evaluations and use a clustering method appropriate for mixed data, such as factorial analysis of mixed data, FAMD. It's particularly valuable for our data set because it can simultaneously handle categorical variables like morphological abnormalities and continuous variables like behavior measurements, preserving the relationships between those different data types. Also, it would be great to develop a supervised machine learning model to predict effective selenium concentrations and behavior responses and to further refine our exposure protocols. And this might even potentially accelerate therapeutic discoveries if we could even make testing more high throughput. These models might be able to identify protective regimens without extensive experimental testing. Third, we'd like to conduct a time series analysis, like Nimay talked about, of our raw tracking data to identify subtle patterns in swimming behaviors, and detect patterns in sequential data like the LSTM approach. So these methods will help us move beyond descriptive statistics to predictive models that could inform both environmental and health specialists out there. So I'd like to thank IDSC very much for supporting this research. I'd like to thank Elvis in particular. He has been an amazing mentor, and he's been very patient with me while I tried to learn Python. And my PI, Dr. Shelton, for her leadership throughout my dissertation project. So thank you so much.
Okay, for the sake of time, let's move on, and we'll reserve the questions for afterwards. Our next speaker is Deema, also from biology. And she's studying how eggs find their polarities.
Hi, everyone. I hope you can see me. I'm a little short in here.
The microphone.
I can. So I'm Deema. I'm a fourth-year PhD student from the biology department. So my topic is an omic approach to unravel the molecular basis for fundamental polarity in animal eggs. So the big question that we try to answer in our lab is, how did the metazoan primary body axis, or we call the anterior-posterior axis, evolve? So basically, how the head is in the top and the tail is in the bottom. So 95% of the animals that have the anterior-posterior axis are bilaterians. So evolving from non-bilaterians to the bilaterians, we can see there's a pattern in apiaxis evolution. So why this apiaxis evolution is important, why we study about it, because it established the fundamental body plan of organisms. So it basically says determine where the head, like head to tail orientation, and where the different organs formation happen. So this apiaxis formation is really important for the success of bilaterians. So when you're talking about and studying about this apiaxis formation, there's a lot of things that we don't know, but something we don't know about is that there's a specific signaling pathway that play a major role in this axis formation, which called the wind signaling pathway. That is the major signaling pathway we study in our lab. So when we're talking about the wind signaling pathway, there's so many different studies that shows that wind signaling need to be switched on right time at the right place. Basically, wind signaling pathway need to be switched on at the vegetal or the posterior part during the gastrulation, and need to be switched on for the in the anterior for the proper formation of the anterior structures or the head formation. So to give a brief idea about this wind signaling pathway, I know it's a little bit complex. Oh, you can see. Oops. So when the wind is absent, okay. Oh, you can't see the, oh, you can. So when the wind is absent, you can see this destruction complex. I'm going to stop doing that. So you can see the destruction complex. So it phosphorylate the beta catenin, which is a co-transcription factor, which move into the nucleus and activate the gene transcription. What happened, the wind present is this wind goes and binds to this ligands, and then it activates the dishevel, which is a very important protein of this pathway, and inhibit this destruction complex. So there's enough beta catenin in the cytoplasm to go into the move into the nucleus and activate just gene transcription. But how this is happening and how the wind knows that where to switch on at the right time and switch off at the one end and the switch off at the other end is something we don't know. So are there any maternal factors, or are there any specific things that inside these embryos that we need to find about? So talking about this, what we think is, okay, let's focus on finding if there are differentially enriched factors in these two poles that we can find. So we focus on doing RNA sequencing and mass spectrometry. So what we did is, oops. So I'm working with C-star embryos. So this is a oocyte. So you can see the big thing inside a little highline. It's called the germinal vesicle. So that determines the animal pole, and the other one is the vegetal pole. So what we did is we did microsurgery and separate this animal and the vegetal halves of the oocyte. So this oocyte is just 180 microns, so they're tiny. And it took me 10 to 12 hours for collect just one sample, like 150 to 300 samples. We had to collect, like once you started, you have to collect it in one day. So I started at the 8, I'm in the microscope for like almost the whole day. So first we send the samples for the RNA sequencing. So to do that, we extract the RNA and quantified it with nanodrop and qubit and make sure there's enough concentrations in each sample, and we send it for the sequencing. And sorry, it's a little slow. Okay. And thanks to the IDSC, I got the access to the Pegasus, and I was able to do my own analysis for the RNA-seq data. So I started with doing quality control and trimming using BBDuck and FastQC. And I did the alignment using the star, and I checked the alignment with the IGV. And I start doing the make the gene count with star quant mode. And by the time more contact us with giving us asking us to give a little update, I was stuck with HT-seq because I want to do like get two different gene counts from like two different softwares just to make sure that what I'm doing is right. And then I remove the low gene count, and make sure to remove the badge effect using the combat-seq. And I did finally differentially express gene analysis using the DE-seq tool. And then once I'm done, I start collecting the samples for the protein for the mass spec data, which is very similar what I did. I collect the proteins and make sure quantified with the qubit and make sure each sample have enough proteins and send it for the mass spectrometry. So once I get, and I did the same analysis with the mass spec, did the DE-seq too, and get the differentially expressed genes and proteins for this from the RNA-seq and the mass spec data. So the A shows the RNA-seq data and the B shows the mass spec data. So from the RNA-seq, I got 73 mRNA enriched at the vegetal pole and 234 mRNA. And interestingly, 91 long non-codings enriched at the animal pole. So this is the first time that we saw that the long non-codings are enriched at one pole. And from the mass spec data, we found 107 proteins at the vegetal pole and almost 1,500 proteins at the animal pole. So when talking about the RNA-seq data, so this shows the Volcano plot and the heat map from my RNA-seq data. So the red color shows the differentially expressed mRNA at the animal pole, and blue color shows the differentially enriched mRNA at the vegetal pole, and green shows the differentially enriched long non-coding RNA at the animal pole. But what's interesting is we found like just two specifically connect with the Wnt signaling pathway, and we were more interested in the long non-coding RNAs, but none of the long non-coding RNAs have a name. They're all uncharacterized. So we don't know which one to choose, and it's going to be a disaster if we have to choose like one by one. So what we did is we are doing a collaboration with UCSD, doing Merfish. So this way we can actually check all 383 genes. So we start this collaboration and we design almost 20,000 probes for these 383 genes, and we have the data, and we started the imaging this week. So we are going to have data like in 2 weeks. What's also interesting is this is the very first time doing this method on the echinoderms, and we don't know how it will go, but hopefully I will get some interesting results. Because this is all computational data, this step is really important to validate what is actually significantly enriched in two different poles. So I'm hoping I will get some, at least long non-coding RNAs, so I can do functional studies and see if there's like any interesting results that we can get from that. And when talking about the mass spec data, again, the red shows the enriched proteins and the AP, and the blue color shows the differentially enriched proteins at the vegetal pole. And we found 15 proteins at the animal pole that have a connection with the Wnt signaling pathway. But one of the most important but interesting proteins that we see in this data set is the custose. So why custose is important? Because it inhibits the canonical Wnt signaling by preventing beta-catenin nuclearization. So the custose, we have just one literature about custose, and it is on vertebrates. And they just check the custose. So this is the very first study for any species that shows that custose is actually enriched at the animal pole, which also explain that the Wnt inhibiting in the animal pole may have like effect from the custose. So the first thing that I did is I tried to do the AlphaFold between the custose and the beta-cat to see if there's a protein interaction between these two. And we saw some kind of interaction, but it's not that strong. But also, we don't have much information about the custose because it's just this one paper. So I started doing the siRNA injections and GFP constructs, so to see if we can see what kind of phenotypes that we are getting. So I'm in the process of doing that. And also one of the important things that I'm interested in doing in this is to see if this custose protein have an interaction with any of the other significant proteins from the animal pole using the alpha pull down. That is something like I'm still stuck in, but mass spec data is like very fresh. I got it in like end of January, start of February. So I'm still in the process. I'm stuck in installing the alpha pull down. But to summarize my data, so I hope this study will help us understand if molecules enriched at the animal and the vegetal pole influence the canonical Wnt signaling pathway, and whether they impact the establishment of the AV axis in C-star oocytes. And fundamental studies will test the significance of the selected set of molecules in establishing the animal-vegetal axis. and their putative roles in activating the canonical Wnt signaling. And with that, I would like to thank my advisor, Dr. Atula Vikramanayaka, and my IDS mentor, Dr. Zhang Wang, and this IDSC Fellow program. And also, I want to give special thanks to Mo, trying so hard to understand our program, the project, and trying to find perfect mentor for us. So thank you so much for that. And I also want to thank my lab and my undergrads who helped with that, and all our collaborators helping with these two projects. And if I have time, I would like to take.
Hello, everyone. So my name is Alejandra Planells. I am currently a second-year PhD student in biochemistry and molecular biology in the School of Medicine. I'm in the Donner Lab. And a little bit of background about me, I study biotech, so I'm mostly a lab scientist. I haven't really dealt with anything to do with like Python programming until very recently, so please keep that in mind. But I do have to say I'm incredibly interested in this, and I do want to take this further, so just wanted to start with that. If I can pass. Okay, so my motivation for this project, I'm going to first start about personally. So I did take Ravi's PIP 792 class on data science for bioinformatics, and I have to say this is when I first actually learned how to program Python, and actually learned what machine learning was, and actually started to learn a little bit about this field, and I saw how interesting it was and how much you can actually do with this. So apart from this, wait. I didn't even say my project title. Sorry. So today I will be talking about exploring peptide small molecule interactions with AI and large language models for versatile molecular detection and biosensor design. So what this basically is accelerating peptide discovery. And why is this so important? Well, as we most likely all know, last year in 2024, there were 2 Nobel Prizes awarded for this, in physics for machine learning and in chemistry for protein structure for AlphaFold. So as you can probably tell, this is a very, very, very hot field right now, and a lot of people are really interested in trying to understand how everything works a little bit more. So moving on from that, how did we make this connection? So in our lab, as I said, we are not super deep into the machine learning field. We're mostly lab people. But one thing that we do specialize in is in biosensors. And what are biosensors? So they're a device that measures the biological or chemical reactions by generating signals that are proportional to this concentration of a compound in the reaction. And this is basically using biological molecules. And such as that, one of those would be peptides. So why are peptides interesting? So peptides can bind to many, many things. They can bind to other peptides, they can bind to proteins, they can bind to small molecules, and they're very, very good for biosensor development. So when we looked at this, we saw that there was already a lot of information on how peptides can bind to other peptides and proteins, but there was little information for small molecule binding. And that is because small molecules are, as the name itself says, so small that it's very hard to actually find anything that will correctly bind to them. And that's when this idea came through to create these peptides biosensors for specifically small molecules. So again, why are peptides so interesting? So they are remarkably specific, and they have robust binding capabilities. And this is because since there are 20 natural amino acids, this means that there are millions and millions of possible combinations that you can do, and therefore different structures that the protein or the peptide in this case can take to target a small molecule. So some of the applications that you can actually do with a biosensor peptide are, for example, you can use it in therapeutic applications, you can use it in diagnostic assays, you can use it in environmental monitoring, and of course in pharmaceutical detection. So we can probably agree that there are a lot of uses for peptides. So this leads me to the next thing. What methods should be used for peptide discovery? So the traditional in vitro method for peptide discovery would be lab. In the lab, trying to test out the peptides and trying to find out their bindings in vitro using in vitro methodologies. This is very effective, but it is extremely resource intensive, extremely time consuming. I can tell you myself, It can take years to even design one peptide because you have to do so many trainings in the lab and use so much material for this. So the other method that there is are in silico methodologies like Autodoc, which are extremely efficient, they're extremely fast, and they can reduce the resource reliance by using in silico methodologies. And one of the examples I was saying is using Autodoc binding simulations, where you will basically select your small molecule and you will use a peptide, in our case, to try to dock to that. This is one of the targets that we were using, a small molecule called BPA. And then we can see how in our case, we are trying to bind with peptides that are 5 amino acids, so a chain of 5 amino acids, and how it surrounds to this. But the question is, we're not just trying to bind one, we're trying to bind multiple. And how many? Well, for pentamer peptides, which is what I was saying, that there are 5 possible spots for them. If we have 20 possible amino acids for each position, and we have 5 positions in this pentamer, this means that we have 3.2 million possible combinations, which is a very, very big number to run. So that's exactly the next point. What happens when you try to run this 3.2 million possible combinations of peptides in Autodoc? Well, I can also tell you, running the simple Autodoc, it would take months and even years. So this was for sure something that is not possible if you want to actually do it faster than in the lab. So this is where our next idea came in that someone in our lab came up with. It's Pepe, who's here today, so I wanted to give him a quick shout out. He came up with a solution. Okay, so what if instead of running the 3.2 million, we run only a small sample size on Autodoc, and then we use machine learning to predict the rest of the binding scores. So with this, we take the peptide library, and from the 3.2 million, we select only 16,000 random samples. And with this, we do our initial Autodoc, and then from this initial screening, we train the machine learning algorithm, and then from there, we can select the top candidates, and we run a second Autodoc, and then from there, continue on with in vitro testings. So an example of what that looks like running a really simple machine learning algorithm is something like this. You have all your conformers, you have your target, and you try to do the docking, so it would look like this. You have first docking, which you get binding scores for this, and then you take this data and put it to a machine learning model. You get a prediction, and then from the prediction, we're selecting the 48,000 best candidates, and we're running a second docking. And as you can see, immediately just from this, we can already see there's a huge improvement in the binding scores. But the next issue that we ran into, and here's where we needed IDSC help, was that we had 2 pretty important bottlenecks. And those were in our in silico screening and in our machine learning refinements. So the first one was the bottleneck that we are using like GBM for our machine learning algorithm, which does not understand context. So I will explain that a little bit later on, but basically using only 5 amino acids, this model can work for it, but if you ever try to go anything a little bit deeper or longer or anything more complex, it will have limitations because it does not understand the context. And that's why our first aim was trying to train a more complex algorithm like large language models for improved scores, which actually do understand what you're inputting and the context of where the amino acid is and its surroundings. And the next bottleneck that we had was the molecular docking timings for Autodoc. So what we came up with was, okay, we are using CPUs. What if we tried to use GPUs, Autodoc GPUs? So this leads me on to the techniques that we're using for the first aim for the machine learning. So as you can see, a light GPM algorithm is basically it's based on decision tree ensembles, so it doesn't really understand the context. At the bottom, you have a graph of how it would basically work. Is it organizes each amino acid, and it basically gives it a score of 1 or 0 if it is in that position. So that's the only thing that it knows, basically in binary, if it's there, if it's not there. But on the other hand, if you use LLMs, it can understand all the context. So you're inputting the actual raw peptide sequence, and by this breaking down, it can actually understand the little codes and the little words of it. And then you can use models that have already been trained, as I was saying, in protein or in text context, and they can understand this. So a little bit of foreshadowing already. We use both text BERTs and protein BERTs, and you might be wondering, okay, why are you using text BERTs? I will go into that later on. But basically, when you're using models that have been trained in protein databases, like our protein BERTs, like ProtAlBERT and ProBERT, they already contain information from like PDB files and all the information that there is out there of protein, so they can understand the context of even the smallest protein to the largest protein. And then text, we had the idea, okay, technically a protein, a peptide is a sequence. So if we try to input this into a model that understands text, could it understand our sequence as text? So this was my first time running machine learning algorithms too. So this took me a long time for just to run this itself. But basically how I did the training was basically you split the data, divide into training, validation, and the test sets. You tokenize, which is basically convert like the raw sequence into token IDs that the algorithm can understand. Then you do a batch loading, and then you do a forward pass to basically pass the inputs through the model for the predictions. Then you do loss calculations to compare the predictions. And from there on, you do some backpropagation, some metric loggings, validation steps. And then you repeat these steps for multiple epochs. And then from there, you can finally get your final evaluation on the test set to assess the actual model performance on the unseen data. So I'll try to go quickly through this. This is basically, I will try to explain everything separately. So at the left, I have a little table of model comparisons over light GPM versus the 3 top candidates of the other machine learning algorithms that I was running. And just by itself, we did want to do the switch into a more complex version because of all that I was saying. There's the more you advance, the more you will need an LLM. So what we first did, of course, we wanted to make sure that the true binding scores fit well with the predicted scores compared to our light GBM model. And as we can see, this one, the still BERT, that was the one that performed the best, was pretty accurate. Then, of course, we tried to look at the R² and the mean squared error to make sure that they were performing correctly. And what we saw was super interesting, that you can see down here at the left, that the still BERT, which is a model that's text-based, was actually performing much better than light GBM, and not just that, but was overperforming the models that were trained on protein databases, which was extremely surprising to us when we first looked at this. And not just that, but it was it improved over 13% what we were getting with light GPM. And then another really interesting fact, if you look to the one on the right on the model size, is that apart from light GPM, which just has the smallest total parameters, the next better one was distilled BERT that has 65,000 parameters, which is a lot, but is less than the other two. So my hypothesis for this is that since we are using peptides, which are way smaller than a big protein can be, the smaller and the more simple the algorithm, the better it can do the task. So, and also a quick comment on this. IDSC was essential for this because running each of these things separate when I was running it like in my personal computer, just each calculation was taking 6 minutes. When I tried running it in Pegasus, it was taking 2 seconds. So just wanted to put that out there. And then quickly, I have 2 minutes left. The results for our second aim, which was the installation of Autodoc GPU. I have to first also say thank you to Elvis because he was also crucial for this. I have to say we tried numerous times ourselves trying to download Autodoc Vina, the GPU version. I know people here have also tried and they were also not successful. So it was great having the help of Elvis and everyone else here. So basically some quick facts about this. Autodoc Vina GPU can run 56 times faster than its CPU version. And from what I've been trying to test, it can execute the ligand receptor binding in 2 seconds, which is much faster than what we were running. So for example, for 16,000 peptides, we were running it in about a little less than a week. And with the GPU version, we can get it down to 8 hours, which means this is 21 times faster than what we were doing. And we're still trying to work on this. We're trying to tweak this. So we hope this can even get better, but this is a 95% decrease in runtime. And so basically this means that in the time it took us to run the 16,000 peptides, we could now run 300,000, which of course would be amazing to also input all of this into our LLM. Yes, basically this is over 1,000 times more peptides. And then if anyone wants to know, because everyone I've talked to about this, they've all come up to me and said how interesting it was and how much they were trying to also use this Autodoc version and they couldn't. So thanks to Elvis, now the Autodoc Vena GPU version is in the Pegasus dev server, and the regular Autodoc GPU is in the Triton server. And some hardships from this, it was extremely difficult for us to set up the GPU versions. We could not have installed it without help from IDSC. And then the initial runs with GPUs, as I guess most of all can know, they're extremely complicated. So also wouldn't have been possible without everyone's help here. And yes, that leads me to the next step, super quick. We want to keep working on this. There's so much more to this. There's so much in this field. In our lab, as I said, we do mostly in vitro lab stuff, but we are now shifting to try to use this pipeline. And this nowadays is our main approach for peptide design for all the targets that we're doing in our lab. And then I also want to continue working on LLMs. I've actually seen an LLM that is trained on not only protein databases, but also affinity scores. So you can actually compare these affinity scores to Autodoc scores by itself. And what I was thinking is maybe I can even get this model and maybe narrow it down to just the small proteins that it's using, maybe less than 10 amino acids, and trying to even train this model ourselves to try to get better scores, because what we're seeing seems that the smaller the model, the better. And then our third aim was collaboration between teams. So as I said from the beginning, we truly believe this pipeline can be used for designing peptides for all targets of interest. This could be for our groups, for other groups, for other disciplines. So we think this is extremely beneficial for everyone. And of course, the use of Autodoc GPU, I've already gotten a lot of interest from a lot of people here at UM. So just wanted to close up with that. Thank you very much.
Amazing. So last but not least, Katarina, Micin. Let's keep it to 15 minutes. All right.
Okay. Can everyone hear me? Okay. I speak kind of low. Okay, yeah. So my name is Katarina Micin. I'm a second-year PhD student on the medical campus. I'm in the Human Genetics and Genomics program. My IDSC mentor was Dr. Abayawardena, who's both part of computer science and also now with HGG. So she's been super helpful. Okay, and so this was the title for my proposal, and this I'll start with is essentially my entire dissertation. So it's not going to be like all the results now. This is like my end goal with the PhD. But it's ultimately looking into the development of a single-cell RNA sequencing guided chronic myelomonocytic leukemia classification model, and I'll get into the background of it. Is that working? Oh, it's still late. Okay. So just really quick background, I'll refer to it as CMML. That's the name of the disease that I work in. I work in a hematology lab. And this is ultimately a pretty rare form of leukemia. It only occurs in approximately 1,000 individuals each year in this country. Clinically, you would diagnose this essentially based off of either blood work or bone marrow samples. And what you're looking at is monocytosis, which is like basically you just see an increase in this type of cell type, which is monocytes. The median age is 65 to 75 years old. So it really affects mostly older individuals. And it is, although it is rare, it is very aggressive. And most importantly, it has a high transformation rate into what's known as acute myeloid leukemia, which is a very aggressive and ultimately fatal leukemia. So for that alone, it's definitely worthwhile looking further into just researching. Okay, cool. It's delayed. Okay, so a big thing with CMML is because it is so rare and because it often becomes another disease, essentially, it's really hard to get any samples with it and trying to catch it really clinically before it progresses. But it is very unique in a few different ways in that it displays both what is called myelodysplastic and proliferative features with neoplasms. And there's really only a few diseases that I have outlined here that share those features. And so because of that, a lot of what we do have for treatment for this is not super specified to the disease. It's more so what we understand with other diseases such as MDS, myelodysplastic syndrome. What we kind of understand works for other things. And we've seen some like mild success with that, but again, it's not super specific and there's only actually 2 currently FDA approved CMML treatments. So this is a huge gap, sorry, and like what is like the currently available therapies for this. And here, I'm not going to get into it, but this is just kind of, if it'll go back, what you see in terms of what's available for the treatment landscape, which you're like, okay, it's a lot of stuff. But again, almost none of these are actually specific to that actual disease and have pretty little success in being not just curative, but even honestly just reducing its progression. So moving forward with that, as I mentioned in the title, what I'm really interested in is developing essentially a new way to classify this disease outside of just what's the traditional clinical classification. And so here are the most commonly used systems today. And again, as you can see, this is really looking at what you would see in a clinical presentation. And so this is, of course, important in that it's descriptive of clinical presentation and more so just disease progression. But I'm interested in also looking further into what is going to be overall prognosis and predicted drug sensitivity, transformation into other diseases, et cetera. Yeah. Okay, so with that, I am a computational student. So going into this, I enjoy working a lot with what is known as single cell data. I'll give a brief overview on that. And I knew I wanted to get more into machine learning. So with that, working with my advisor, I thought that it would be pretty interesting and beneficial to essentially try to work towards combining those two. And so the ultimate goal is to develop a classification model that uses this specific type of sequencing data as the ultimate input. And from there, it's obviously a big undertaking, but be able to get from that data certain descriptive features that can then be used to inform more descriptive classifications. Okay. Should I go? So I don't know if everyone's familiar. I'm gonna give a very brief overview. So what I work with as my primary technique, I don't do the wet lab, but this gets to me in terms of just like files, is that we do single cell sequencing. 10x just refers to the company that we use. It's pretty like standard for this. And essentially what you get, like someone else I mentioned, they did RNA sequencing. That would be the bulk version of what this is. What you get is still RNA readout, but at a literal singular cell level, which is extremely high resolution. And what you ultimately come out with is gene expression data per cell inside whatever sample that you're working with. And so this is really cool because bulk RNA is also very informative, but ultimately is an aggregate version of this. And what I'm looking for again is classifying features. So there's no like higher resolution than what this offers me. Okay, cool. And so what I ultimately work with is this part of that, which is just all the data analysis. Kind of what I was doing with what was relevant with working in this fellowship is just finished essentially automating the initial steps of what the overall processing is in analysis, which is a lot of steps. So I don't include a lot of graphs because there'd be a lot of it, but you do a bunch of things just to get to even a starting point into trying to get anything from this data. So I won't go through all of it. Some people have kind of mentioned that they've worked with some of these things. So you do all of that when you're working with this type of data. This schematic that'll show up offers a very condensed version of what you do to work with this. It essentially is because I ultimately work per sample with thousands of cells. Typically, you want to aim for at least 5,000 cells per sample that you sequence. And obviously, to drive any algorithm, I'm going to need a lot of samples for that. So you can imagine how big this data gets. And so what you essentially do post just pre-processing like any other data set is just a bunch of different dimensional reduction methodologies, essentially. And so I won't get into it because they're pretty late, but like I do a lot of this. It's just a bunch of that. Cool. And so starting off with, the big thing is just, okay, how are you going to get this data? My PI is a practicing hematologist, so we do have access through our own cancer center, the few patients that we see. But again, this is also just a rare disease in general, so it becomes difficult, and then they become just very precious samples to work with. You have to get bone marrow samples. It's all very like difficult. But from what we source from here, I started off working with just basically 4 samples. And that's how I kind of got into like the swing of all of that. But then for my actual like dissertation, just a lot of like publicly mining what's available. And like so far I've found almost 80 samples that I'd be able to work with. And that's just with like more like very just like shallow digging. So I'm sure I could find more. So I'm not too much worried about finding stuff that could drive that. And so here I'm just going to show for those initial four samples what this analysis, what the ultimate output essentially looks like. And so this is a UMAP, a visualization. And so what you're seeing here is after I do all of that pre-processing and analysis, the main goal is to be able to identify for each cell, like what type of cell is this? Like what are we seeing in that sample? And so this is again from a bone marrow sample of a CMML patient. And so I don't have a cursor. What you see in yellow, or no, sorry, blue, is a monocyte population, which I mentioned earlier is the clinical feature of this. So that checks out. But this is essentially from all those patient samples merged into that. And so I show this just to also show that you ultimately see a lot of distribution even within the actual, patient samples, even though that's what the big picture looks like. If you look at the actual breakdown for each patient, they all have very different distributions of those cell types. And so going into this, I already knew that this is going to probably be one of those like really classifying features. And this shows it as well. This is like, again, just that's the merged on the very right. And then what you see here is I'm just actually splitting what each patient's actually contributing to that. And so I do a lot of that essentially with all these samples. What I'll show is just some of the initial steps of working with a bigger data set that was publicly mined from this paper indicated here. I got it off of GO. And I'm working with a total of essentially 39 different patient samples. And so this gets up to already hundreds, like almost 100,000 cells level. So everything that I do from here is me working on Pegasus or Triton or other supercomputer access that I had, which was, I think, the biggest part with working with this fellowship is just me having the actual computing power to do anything. And so once I did all of that other stuff that I mentioned earlier, this is the final clustering that I've decided on now. A big part of it is also just trying to find out what are real clusters. You have to mess with the resolution and like your k-means, it's a bunch of stuff. But I ultimately ended up with 135,000 different cells. And the big thing now is finalizing annotations, which I've spoken with other people here about. This is the predicted annotations that I'm working with now, but again, as you get to bigger sample sizes and the number of cells, it becomes very difficult to start understanding really what's like at play here. And again, this took a lot of computing power because what I ultimately do is just a bunch of different calculations to find differentially expressed genes within each cluster in comparison to each cluster within the cluster. It's just a bunch of stuff. And so I'm going to do that for all those data sets. I don't have all the data here, but that's okay because we're pressed on time. And I kind of sped through it, but this is like my ultimate PhD is to find out which are the best combinations of different machine learning models that essentially will give you those classifying features that I'm most interested in. Yeah. So short term, just like finish the analysis. Long term is basically finish my PhD and do everything that's involved. And like I said, this is like my main project, so it's kind of hard to give an update on. I work on a bunch of other side projects too that I love to talk about. I do also like docking stuff and like drug stuff, other things. I'm fully computational. So the big takeaway for me was also the mentorship of someone that's even more advanced in my knowledge of this. And also, again, just knowledge and like what's accessible in terms of like computing power is a big thing that I'm always worried about. Like, how can I run all this stuff in a more efficient way? So that's been really great. Thanks to my lab, that's us there. Thank you to all of IDSC, my mentor. Yeah, okay.
Great.
So thank you to all our IDSC fellows, their mentors, everybody that helped along with making this possible. Nick here has been a stalwart supporter of this program and to reaching out to the different groups. One thing that I would like to say, that this is not the end all of your interaction with IDSC. We just opened the door. And so please continue working with IDSC and with your mentor. That's the goal here. Sort of, Alejandra just gave the example of someone who wasn't really trained in computing, and now, the whole group is open to it. So please continue working. A lot of you are actually just sort of, you have a long horizon in front of you, so please make use of it. The second thing that I noticed, particularly in this year, that a lot of you were sort of needed to install software to speed up your calculations, and that was important, and that took a little bit of time to articulate the need, actually. I would encourage you to use the knowledge, software and hardware and system administrator to keep on using IDSC as your central computing platform. And with that, I'll open it for questions.
And snacks. And snacks.
And snacks. Oh, and snacks, please, help yourself. Nice.
That's very good.
I'm Michi.
So Ken, like for you, like, so I know that students are able to like apply to like create projects using IDSC resources. Those usually like have a time limit on them, or how often do those go through? Things like project requests.
That's a question for Ravi, but
Question?
I was just, I didn't realize until like earlier this year that all UM students can apply for computing resources through IDSC. So I didn't realize that. And I think that's a really cool resource that maybe people don't really know exists. So if you get, let's say you apply for a project on Richter or something, is there like a time limit?
You usually get an allocation.
Right, okay.
Until you exhaust the allocation.
Okay. Yeah, okay, sure.
Yeah, it's usually depending on how, other modeling people would call it exhausted like that. But usually,
Okay.
But then your advisor can ask for an allocation,
Sure, okay.
right? And then allows the group to have. So any student, no questions asked, can get an allocation to learn, to train, to do that. So if you go more than that, then it's part of a project that then your mentor is asking basically to get access for you to do your project.
Sure, okay, I was just curious, Ben has his own like computers and stuff that we like all use in our group, but like, I just thought that I just didn't know anything about that, and I wonder if a lot of students don't realize that.
That's absolutely true. Maybe they don't.
Yeah.
I have that. I got 90K units on last year, January, so I'm still using it. I just used like 30K for now, so I still have a long way to go. But I think they just give, so when I get it, they said like it's just for 1 year. So before end of this last year, I just email and ask. They said like they can renew it for another year. So I'm not sure. Able to. So
I'm just curious.
but the more incentive for you is to bring your problem and then translate it into an application. Take it forward.
If your group is thinking in investing in hardware and software, you're sort of better off leaning on IDSC to do that. They have the know-how for the system administration side, and it will be a shared resource. The group doesn't have to bear the cost on its own. Like with Autodocs and Alpha pull down, they can be difficult to configure to a specific system, specifically when you want to do big problems, right? That's, to do something new, you have to do big problems, because if it were small, somebody else has solved it. So. So anything you guys can tell us for next year? What would you wish to have done differently would be nice.
A little more time.
Yeah.
Just a little more time. I feel like I could, I feel like, so what, it was like 5 months, pretty much, 6 months. I'd say 7 months, 8 months. I feel like I have been very useful for me anyways.
Okay. Well, you know, this is not the end of your collaboration with
Oh, no,
Sam.
certainly not.
Are you going anywhere, Sam?
Yes.
When did you submit your applications?
October, I think.
Yeah, so it's normally we do it, as part of a semester, right? Because I mean, for graduate students, semesters are not that important, so to speak, right? But for undergraduate, a semester is, a kind of a unit that you organize around. So, and because we have both graduates and undergraduates, we actually do it for a semester. And as Mo was saying, is that, this is just the initial engagement, right? So don't expect that you are going to get all the project done in one semester. Would it be nice, but
Sure. It would have been, I mean, I feel like it would be cool to be able to, like, for my capstone presentation, to really do more for the capstone presentation, the more
Sure.
time. That's about it.
You already went through a computational challenge, which was to configure your CNN LSTM. So that's a big step.
That was like half of the time.
Well, but also this is also the part, right? That, in order to start getting more and more advanced in this computing, it's not like, you are just using your laptop or your desktop, right? So you have to spend the time to understand what it is and all of these kind of things. And I think this is also part of, this internship, to understand how you can scale up, to work with bigger, computers, what it takes to do these kind of things. And of course, it's not that simple. If it was that simple, we would all kind of, have done this long time ago. So this is also part of, the idea of what we have these fellowships, to get you familiarized and what it takes to work with larger systems, that it's not your desktop. Right. So it's, I mean, I agree with you that it's nice to have always to have good data to show, but don't forget that the journey is also part of that. That's why you do a PhD. That's why you do a 4-year, degree, right? So I keep point well taken. I mean, we will see what we can do in terms of, add some more time, but
Well, your argument's pretty strong, so I can see that.
No, but I would like to stress that this isn't supposed to be the end point of the collaborations. We hope to keep it going in the future, especially if you're here for the next 2-3 years, whatever.
Yeah, definitely. There's a lot, I mean, there's a lot to do, there's a lot more to look at, so.
Well, I have a question for you. How did you find out to apply for this internship? Because I think one of the issues that we want to do better is, as you mentioned before, is that how we can, reach more people to understand, and more students, that they have this kind of, opportunity. So how did you find out?
I had seen it through an email.
Yeah, email, like 2 years ago.
My PI.
Well, I taught Nimay and I plastered it in front of the class.
We're just trying to find the most efficient, the best ways to reach you guys, right? That's what I would try to say. So, and we're trying to find, I mean, there's not a silver bullet. That's one method that it catches all. But we're trying to understand how, we can be more effective to reach, more of your colleagues, in the years to come.
The suggestion I have, at least for Rosensteel, is like we have town halls, we like student town halls. I think that it'd be cool if someone from IDSC, or even if like the fellows from you before came to like the
That's great.
orientations and stuff, and just like gave like a little presentation, at least like plants to seed, like, hey.
I mean, that's something that, yeah.
I think also I'm on the medical campus and like I said, I'm a fully computational student, so just by like it just became relevant to me to know about IDSC, and so I just knew about this and I looked into it. But I do think sometimes I even just mention like the word IDSC to other colleagues who are like wet lab people and I don't even know like, it exists. So I don't know. Also, there's like physical distance because I know you have a computing stuff, computing, but a lot of the stuff is on main campus. And I think unless like my other fully computational like cohort, we know about IDSC because it's just like relevant to the work we do that we need some type of like exercises to try and. But at least in my medical campus, I would say so.
So what would be a more effective way
Well,
for
like
the,
we don't do like town halls per se, but we have like our like student government stuff, but we host a lot of like things.
Yeah.
I don't know, just like a physical presence somehow at some point. I think.
sure,
I talked to Dana today.
Yeah, he's like.
We are kind of scheduling a couple of seminars on the medical campus. As a part of the ACS outreach.
well, because we always do some, workshops and so forth. The question is that how we can be more effective to get, so we do, software carpentry on the medical campus and so forth. So the question, we try to find, again, how did you get approved? How did you know and how we can do things? The point well taken, more kind of, we need to reach more of kind of the functions that you guys do as students and so forth to get informed, to at least give us 10 minutes, somebody to come and say a few things. And so
Maybe
on.
just like sending emails to like some of the like notice listservs for some of the different campuses, because I honestly didn't get IDSC emails until I signed up for that listserv. So you can send an email to like the notice at Rosensteel listserv, and that goes to everybody at Rosensteel, so.
Okay, more broad.
Well, but
But
this
that's
is
like the-
controlled, this is where the issues come. But this is controlled by the school
Sure.
or by the college or whatever, right? So, and they have, so it's not, we cannot send that. So I just want to make sure that you all understand some of the issues.
I
So
do
you have to be.
I understand.
So, but that doesn't mean that we should not, try to do that more targeted, right? So.
Or even asking some of like, so if I were to send, like asking the previous years, like fellows, like if I were to send like a notice or something to Notice Rosensteel or the info, let's serve, I could just ask Christy, hey, can you just put this through?
Sure.
And
Sure, that would be
she
great.
do that, so, especially coming from a student, I would imagine.
I got to know it from my
Maybe different
PI.
faculty members can send.
Yeah, I got to know it from my PI. So he basically sent the email to all our lab members
Right.
and asking us to apply if we are interested.
Mhm.
I feel like that's a nice way too. Because we didn't get the emails first. Yeah, I got the access to the Pegasus, but I did not get any of the emails until I sent the email to the IDs asking, can you include me into the listserv?
Investigate.
Jessica, how did you get
Email, so.
email?
I think it was just like a campus-wide email, but it was kind of buried in like mid-text. So I think if I had scrolled really fast, I
Yeah.
probably would have missed. Yeah, something
Yeah.
like that, so it wasn't.
And so you go there
from
like
IDS.
1-to-1, you
And I
don't
also
see it.
wasn't subscribed to any emails.
You should do a better job. I think Alejandra, you learned about it
Yeah,
from
through
Robbie,
Robbie, but
right?
I also saw it in your emails, but I wasn't in your listserv, so I did get it through that. Through the emails. But I also have to say, in the Med Campus, we sometimes have like, I don't know what they're called, but basically the people that are in core groups and core labs, they come and they do like a little setup where they each have their like little stand about their core lab. And I know a lot of people go there to see what resources the university has. So maybe it would be interesting to go to those days where they present the core labs.
The tabling.
Yeah, like offer it as a service.
So actually, well, this is a question for you, Katarina and Alejandra. Will you be our ambassadors to these events?
I'd be happy to, yeah.
Nothing works like word of mouth.
Well, I mean, I have to tell you, I mean, you tell him, but if we look at all the IDSC Fellows that we had over the years, I have to tell you, at least we have at least 2 or 3 from the medical school every year.
Yeah, I think there's also like.
I mean, but of course, the medical school is massive, right?
Yeah.
But we have at least 2 or 3 every year from the medical school. But again, what I'm hearing is that we are still, not reaching the majority of the students. That's what I'm hearing.
Or I think students who aren't already like computational, like, or at least parallel, like maybe there's someone in their lab already kind of works with like. Cause I was thinking about the US earlier, I got that like advanced computing student grant before I applied for this, which got me like service units for Pegasus to try it. But like, it was like pretty easy to apply for, to like look for it. And when I would tell people about that, they were like, "What are you paying?" I'm like, "Oh, nothing, because I have this." And they were so like shocked by that being a thing. So I think I heard it from the guys, I think actually maybe exists with one of his programming classes, but I was already involved with it. You know what I mean?
No.
I only hear from other people that are already like buying from this or something.
Yeah.
So
I want to ask a quick question to all you guys too. So I run the software carpentry workshop and we do them in the CTSI training center. Have you guys heard of these before?
I have. That's how my lab members take them.
Okay.
Yeah.
No.
Software carpentry workshop.
No.
It's like ours.
It's the way she sends through an email.
Rasmus has a student listserv, like a grad student listserv,
Oh,
right?
yeah.
Okay, I'll get them on there.
So we can get medical school, but also I want to hear from other schools. So for example, what I see here, for example, this year we didn't have anyone from engineering, for example. But yeah, I mean, yes, yeah. All of their, including music school, by the way, believe it or not, they're doing a lot of great things there.
Yeah, one of the big steps is to transfer from using your laptop to using these big machines. It's not an easy step, and your programming skills have to kind of also do a jump. I hope they have.
Yes.
I'm still looking for someone that has like a foolproof way of like identifying like let's say just 3-H resources. Someone tell me, like, I just wanna like plug those numbers in and like to like some equation that it gives me like the perfect client. Like desk client that's like, I don't know, like.
And the easy button
cluster,
at the
like,
same
a good
time.
cluster
right?
system. Yeah, I mess around with that so much, I just love that tool.
Great.
All right, thank you all.
Thank you, guys. Take care.